Hi. Today I want to continue with the formation of the protein and peptide. Protein and peptide in the cell, they can produce in the process or mechanism which called transcription and translation. Okay? Inside the nucleus we have a DNA. Okay? It can produce mRNA, which called process transcription, which send information of the DNA with the mRNA group to the ribosome. Okay? It has a codon and which can bind to the ribosome. Ribosome has the two parts, a small subunit and a big subunit. In the big subunit it has a 2-3 place which can tRNA bring amino acid for there, and tRNA transfer amino acid which bind to the amino acid, bring amino acid to the ribosome. Also, ribosome has the rRNA which has the information for the process of the production of the protein, and they can bind each other and make a peptide bond. From the biochemistry, also we have two types of the ribosome. The ribosome, which is in the rough endoplasmic reticulum, they produce mainly amino acid which needs in the outside of the cell. Mainly we can find in the glands, for example, in the pancreas, on the liver, and also we have a free ribosome, which is in the cytoplasm. They can produce protein for inside the cell for use of the cell, because they can produce cytoplasmic enzymes or proteins. When we want formation of the protein in the ribosome or in the laboratory, we can say we first amino acid, okay, bind to the second amino acid, and dehydration happen. Okay, the carboxylic acid of the first one with the hydrogen of the second one produce water. And the new bond formation between two is the peptide bond or amide bond. This bond is the peptide bond. Okay? This part, the peptide bond, is very important, and we can continue these groups from here, from the carboxylic acid part, and continue the structure. In this case, the structure will be like this. We start with the amine group, go to the other amino acid, second one, go to the third one, and continue until the last one. For example, I can say it can be a number, and the last one will be this structure. Then the peptide or protein structure starts with this amine group, we say N-terminal. And finish with this carboxylic acid, we call this the site C-terminal. Okay? And the isolated point of this peptide depends for which type of the amino acid they have. If have a more polar acidic or polar basic, the pH can be different. And if we have a other amino acid, maybe slightly acidic or basic, but mainly depend for the type of the amino or where we can find acidic or basic or neutral or polar or non-polar side chain. Okay? As you see here, this big amino acid side chains, sequence is called primary peptide. primer peptide, it's like rope. It starts with amine group and finish with carboxylic acid group. As you know from chemistry, the sigma bond can be rotated easily. Then this peptide bond can be rotated. But the amide bond or peptide bond cannot be rotated because, as you know from the chemistry, it is a peptide bond, if you remember. Okay? We can have a mesomeric structure. It has a minus K and it is a positive K. The structure will be changed to this structure. Okay, the amine bond will be double. And this double bond cannot be rotated. It's very important, you know. This is when they start rotation, okay, the amine group and carboxylic groups can be affect each other. Okay, for example, I will show you. This is a like rope. Okay. For example, as you see here, I draw. I have a big primary peptide. Okay, start with NH2 and continue to COOH. Okay, it has a sometimes rotation happen between the carboxylic acid and the fourth amine group. The fourth amine, this amine, has a hydrogen bond. Okay, for example, I have a first amino acid start and goes until the fourth one. Between the first and the fourth amine group, you see, there will be hydrogen bond. It will be alpha helix. Alpha helix, it is a peptide, okay, which has a hydrogen bond, and the hydrogen bond will be between the first amino acid carboxylic group and the fourth one amine group, and it's continuous because it's when it's rotation. Okay, for example, this peptide is rotate, and when it close, for example, it's like this: close each other, the first and the fourth will be a hydrogen bond, and it will be more stable. It is more stable. Sometimes it will be it's in like this shape. We call beta sheet. Why is the beta sheet? Okay, because for example, start with the NH2, COH, or COH is a parallel I wrote, NH2, one amine group with one carboxylate. It is, I draw again here, NH and the CO. It is a hydrogen bond. It will be like this. Okay? If it is between the first and fourth, this hydrogen bond will be alpha helix. If this is opposite each other and make like a paper or like a sheet, we call it is a beta sheet. It's very important. Okay? We will have other also structure about the secondary structure is very important for in the biochemistry. And as you see here, they have just hydrogen bond and primary structure with the peptide bond. Sometimes, in the beta sheet also can be have a parallel and anti-parallel when the carboxyl NC terminal in the same side. In this case, will be parallel. If the opposite side will be anti-parallel. Sometimes they have a different type of the peptide. For example, alpha helix with the beta. We call this super secondary structure. You see, it is these two are the secondary structure. It is the primary, as I wrote here. They are the secondary structure peptide. But sometimes we will have a difference with a super secondary structure. For example, we will have a just a hydrogen bond, but different case with more than one group. For example, if we have just beta helix, alpha helix or beta sheet, it is just secondary. In the super secondary, for example, when we have, for example, two beta with one alpha. Okay, for example, if I have a two beta, for example, parallel, I have this beta is the beta. Okay, like it. But in the middle we have alpha also. Okay, by each other. These are beta, alpha, beta. It is super secondary. You see, we don't have other like this non-polar bond. We don't have a salt bridge. We don't have a disulfide bridge. We don't have any of them. We have just a secondary structure and with different structure, with more than one. For example, we have a 5 or 6 different beta helix, beta sheet. For example, it calls Greek K, or yeah, we can say other. We have a beta parallel barrier because it makes more. It's like a make a hole in, or it can make a membrane, protein, and we call beta barrier. And this is they called super secondary. It's very important. What is the difference of the secondary and super secondary? It is about in the secondary we have just, for example, alpha helix or beta sheet or loop, but in the super secondary we can have more than one alpha or more than one beta. They mix each other and we form a different structure. Also, we can say modify. Okay. Now, if we have a very big primary peptide, very big, okay, and this peptide has alpha helix, beta sheet, and the loop, okay, they have it just hydrogen bond, okay. I draw very fastly, and I show how is it possible, and it will be very nice structure. I will explain it. As you see here, for example, just a minute. I have a very big structure. For example, I have this structure. Okay. Okay? It's a primary. And it starts to rotate. And for example, the first part will be start formation of alpha helix with hydrogen bond. Then one loop, then start the secondary bond. It's a secondary sheet. Again loop, then again alpha helix, okay, and again one loop, one secondary with the beta sheet, and that's it. Okay, as you see here, this primary structure change. This part is the alpha helix, loop, beta sheet, loop, alpha helix, loop, beta sheet. It will be the structure. It's a secondary. Okay, they are freely they can freely move. Okay, all of them. Okay, for example, here I have one cysteine. Here I have one cysteine. As you see, we studied in the classification. Here I have one cysteine. Here I have one cysteine, 4 cysteine. Okay, when they have 4 cysteine, when they start to move to each other, between them make a disulfur bridge, and they lost the hydrogen, and they bind each other. Okay. Now, this alpha helix cannot freely move. They make one bond with this beta sheet will be a little stronger, and this one also make a disulfur bridge between them. Okay. Then between the 2 cysteine we can have a disulfur bridge. Okay. It's not. Now these 2 parts is fixed. Okay, other parts will be slowly closer. For example, here I have a aspartic acid. Okay, for example, here I have aspartic acid, COOH, CO minus, and here I have a lysine, NH2 most positive, NHC positive. Between them can be formation of the salt bridge. Then I write here between cysteine and cysteine, it is a disulfide bridge, between COO minus carboxylic acid group and NH3 plus can work the salt bridge or hydrostatic bond. Okay, now it is also fixed. You see here, I have here COO minus, here I have NH3 plus, the also bond. Now more stronger, as you see here, it's more stronger. Okay, and if they are, we see more close to each other, but here, for example, I have a valine. Here I have a alanine. Okay, here I have a leucine. Okay, 3 non-polar cycles. Between them also can formation of the van der Waals bond or hydrophobic interaction. Hydrophobic interaction. This hydrophobic interaction is very important, the proteins in the membrane, the transmembrane proteins, because the hydrophobic parts can bind to the phospholipid bilayer in the middle side and prevent the passing of the polar molecule from the other side. And in the inside of the peptide, they can make a hole with the polar amino acid, which can, for example, it can make a canal and ions can pass very easily from this side. Okay, as you see, this non-polar also can. The third one between the non-polar amino acid, they can make a hydrophobic interaction. Okay, sometimes there is also interaction, new hydrogen bond between the polar compound, for example, between the OH groups or with carboxylate or other compound. This is structure called the tertiary structure. And as you see, they are more stable. They have a disulfur bridge. They have a salt bridge or hydrostatic bond, and they have a hydrophobic bond, and also they can have a new hydrogen bond. They will be the tertiary structure. Okay, as a tertiary structure, if we have several tertiary structure, for example, they can have a domain part. Okay, for example, we can have a quaternary structure. We can call the functional peptide or functional protein, and because in the, for example, it is domain and also this, and the structure of the protein is very important in the function of it, because for the binding of the ligand or other peptides or drugs or everything, we need a spatial shape and structure. We need the active site from the enzyme, or we need a spatial canal form in the transmembrane protein, or all of the protein has a specific folded structure. We will see in the next part, in the next lecture about this, and I hope it will be good, and I will see you next lecture. Have a good time.